recording. Perfect. Share. Lots of technology. Okay. Here we go. Zoom, you guys can see the PowerPoint. Awesome. You guys also will have access to that on my Wartburg. All right. So does anybody know what's going on in this picture? What's going on here? Has anybody ever had this before? Yeah, Sam. Yeah, drawing blood. So Zoom people, Sam just said they're drawing blood. Maybe most of you or some of you have had your blood drawn before. Maybe some of you have donated blood. Maybe you've donated plasma. We'll talk about how that applies in a little bit. You'll see that some of the PowerPoints that are created by the textbook have some very interesting pictures in them, and we will find more of those throughout the year. So here's those points to ponder. I don't need to read through those questions, but they're just, you guys can't necessarily see it all, can you? Okay. You guys can't really see that last bullet point, but I'll make sure that you guys know what it says. Must be a setting thing. I have no idea. Thanks for the little technical difficulty here. Photo viewer. Set up? Yeah, set up. Auto run. Yeah, that's not what I want. Anybody really proficient with these things? Okay. Let's do this. Turn it off. Sorry, everyone. The PowerPoint wasn't working in class. You'd think it'd be harder to do it online than it would be in class. Okay. We're going to keep going just a little bit so I don't waste your time. Like I said, those points to ponder are just the beginning of the PowerPoint. It's a good way to refresh yourself for the exams. Those are some good questions. If you can answer those without looking at the bullet points to answer, those are some pretty good exam style questions. I wouldn't say that I literally take them from the PowerPoint to the exam, but maybe there's some twist in wording or a little bit more depth in the understanding, but you can start to see those on that points to ponder. Okay, so the next slide, which hopefully you guys will see in a little bit here, it's building blocks from small to large. So matter, elements, and atoms. So we've talked about a little bit about how things are organized from atoms to the biosphere. Atoms are those super itty bitty molecules that you can't really see at all with your plain eye, and you'd have to have a very high-tech microscope in order to see that. And some people have the capability of doing that. Matter, you maybe learned in your high school biology or high school science classes, anything that has mass and takes up space. So this table has matter, this chair has matter. It's a part of what makes things that take up space and have mass. An element is a basic building block of matter that cannot be broken down by chemical means. So maybe you've seen a periodic table. My classroom last term had a periodic table in it, but a periodic table is that, we're back to here, I don't know what to do. Computer. Picture. Anybody good at this stuff? Yeah, you want to try? There you go. Okay, we're going to work on that. So elements, like I said, those are the components of the periodic table. For those of you on Zoom, we're having a technical difficulty with the PowerPoint, but it's okay. We're going to keep moving along here. So atoms are those smallest units of the element that retain the element's physical and chemical properties. These bond together to form molecules. So when you have, okay, yeah, so some elements. Anybody have an example of what an element would be? Anybody know any of the elements? Anything from the periodic table?
Iron.
Yeah. Ooh, what did somebody on Zoom say? Yeah, go ahead, Zoom. Was there somebody? You guys heard somebody too, didn't you?
I think Barbara said iron.
Iron. Yeah. Does anybody know the letters for iron?
Fe.
Fe. Yeah. So that's an element. Anybody else have an example of an element? We'll do maybe one or two more. You had one, Sammy, and then we.
Carbon.
Carbon. Yeah. Do you know the letter for it?
Ca.
I think somebody said CA. You know, it's okay if you don't know. It's just
Ca.
it's just a C. So carbon is just a C. So let's look at some of those on that next slide. So this is a very condensed version of the periodic table. It took out a lot of the elements in the middle. Nick is working on our PowerPoint here in person. There we go. Thanks. All right, so the slides that the people in person missed was this one. I'll leave it up for just a second if you'd like to look at that. Okay, so we're talking about elements making up the periodic table. The people on Zoom are looking at a condensed version of that periodic table. The bottom or the columns are the groups. So when you look at a periodic, ooh, there's a cord there. There's so much going on. Okay. So you have a periodic table. It's going to look kind of like this. It'll eventually have different columns. These are considered groups. And these are considered periods. All right. You guys have access to this online, so I'm just going to push it forward so you can see this. All right, so very condensed version of the periodic table. The different terms that would be really helpful to know are at the top: anatomic symbol, anatomic mass, and anatomic number. So when I asked what was that letter, that would be your anatomic symbol. So Fe or C for carbon, that's your anatomic symbol. Okay, the anatomic mass, does anybody know where that is found? Maybe you can take a peek at that picture. Yeah. Under the yeah, under the symbol you can find the anatomic mass. That's the number here. A lot of them happen to have decimal points with them. It's to 2 significant figures in this particular periodic table. Maybe you guys know what significant figures are. That's just that they're keeping these all the same. The numbers on the bottom. In other periodic tables, they may have different significant figures. So this may be an estimate a little bit, but we don't really matter. It doesn't matter to us as much as that we know where we can find that anatomic mass. And the atomic number, does anybody know where to find the atomic number? Yeah, so that atomic number, as you can see, it's right at the top. And what this does is it's just for organizational purposes, but also in a little bit we'll see how that atomic number plays out a little bit in our favor for organizing elements. Okay, so moving on. Good. Everybody's got the subatomic particles of atoms. So you've maybe heard of neutrons and protons and electrons. I was thinking the other day, did anybody ever watch Jimmy Neutron? Yeah, when you were a kid, I was thinking about that the other day. I'm like, oh, that's why his name was Jimmy Neutron, because of neutrons, but I didn't really get that at the time. So neutrons are neutral, and that's really helpful that it's in the name, pretty much, right? The word neutral and the word neutron both have the base word of neutral. It's uncharged. So when things have a positive or a negative charge, positively charged things are protons. Negatively charged things are electrons. Neutrons just don't have a charge. They're neutral. The neutrons and the protons make up the nucleus, and we'll show a picture of that in the next slide that'll help you understand that a little bit. But what I think of, and hopefully maybe some of you can see this here. So when I think of an element, I think of the protons and the neutrons making up the nucleus, and then, excuse me, these have to be zeros. And then the electrons are going to be orbiting around the outside. Okay, so hopefully maybe some of you had some basic chemistry and understand that a little bit. This only works for this particular one here. All right, so I have 4 protons are positively charged, and I have 4 negatively charged electrons, okay? And this would be a neutrally charged element. Okay, so here we have subatomic particles of atoms. So we have different particles here. We have those protons, neutrons, and electrons, and what their charges are and their atomic mass unit. Oh, we know what the atomic mass is, right? The atomic mass was that number on the bottom on the periodic table for that specific element. Okay, so we know what the atomic mass is. Maybe that has something to do with the amount of protons, neutrons, and electrons that are in there. So let's see if that's true. So this is figure 2.2 in your book, and it says the atomic structure of select elements. So here we have carbon. All right, that was an example of somebody who said that was an element that they knew. And if we go back, if I'm able to do that, all right, so here is periodic table. Carbon is right here. It is in group number 1, 2, 3, 4 on this periodic table here. And it says it has an atomic mass of 12.01. So let's go back here and see how that works out. So it says that there are 6 protons and 6 neutrons, and then there's 1, 2, 3, 4, 5, 6 electrons around the outside. Okay, so then we have nitrogen. Let's see how that works. Can anybody guess what the atomic mass of nitrogen would be? Should we just go back and check? Let's go back and check. All right, so nitrogen, it's right here, 14. Okay, so we can we don't need that 0.01 as much for this question, but the atomic mass of nitrogen is 14. And we see that with 7 protons and 7 neutrons. Same thing with oxygen. Can anybody guess what the atomic mass of oxygen would be? 16. Yeah, you guys are kind of getting it. And here it is at 16. And then this PowerPoint here, you guys can see it here. The atomic mass of oxygen is 16. All right, so what does that mean exactly? That just means that we can add the protons and the neutrons to get the atomic mass. Okay. Let's move on to isotopes. All right, isotopes are the atoms that have the same atomic number but a different atomic mass because of the number of neutrons differ. Okay, so we just said that atomic mass, we add the protons and the neutrons together. But these isotopes, they have the same atomic number and a different atomic mass. So there must be something off there, right? So the atomic number was the number on the top. Remember in the element box, right? We had the atomic symbol, atomic number, and the atomic mass. So they have the same atomic number, different atomic mass. Radioisotopes are useful in dating old objects, imaging body organs and tissues through x-rays, and killing cancer cells. Radiation can be harmful by damaging cells and DNA and or causing cancer. So you've maybe heard of like radiation therapy or x-rays, you know, that's pretty common. If you ever break a bone or things like that, you'd get an x-ray. That seems to be fairly common. So radioisotopes are going to be these elements that are isotopes that are radioactive. So let's show an example of what that looks like on the next slide. So this is the difference of what we can do with isotopes and radiation and imaging. So it says these are some medical uses for low-level radiation. This is figure 2.3 in your book, okay? So here, this is just a little part of a certain organ, and that organ happens to be your thyroid gland. So your thyroid, does anybody know what your thyroid is kind of important for? It's okay if you don't. How about zooming at people? Does anybody know what your thyroid is important for?
Is it metabolism?
Yeah, metabolism's one. It also houses a lot of hormones. But yes, through hormones, it does help regulate your metabolism. I've known people who've had to get part of their thyroid removed. It's just because probably, like I said, varying hormone levels, and Macy was right, it does help with your metabolism in different ways, okay? So with the radioactive isotopes, we're able to take an image of this thyroid to notice that there's a missing portion of that organ, okay? Here's another example on the bottom in B. Can anybody guess what this is? Or has anybody had something like this before? What's that bottom image showing? Does anybody know? Yes. What? Yeah, it's a CT scan. So we're able to see some activity of the brain. There's been studies that have been done where you can, you know, stimulate different parts of your body and you can see it react in your brain. So people will do this to maybe look for an injury, a tumor, things like that. You'll be able to see some abnormalities with radiation. So it's a really important part of medicine and understanding those things. All right, so molecules. We're talking about molecules. These are made up of atoms that are bonded together. They can be made of the same atom or different atoms. Okay, so an element is different than a molecule. I'm gonna need to grab an eraser, which they don't have for me. I wonder if this is my new eraser. Do you guys think this is my new eraser? Yeah, I think this is my new eraser. Okay, yeah. Okay, so what is a molecule? The way I think of it is that a molecule is made up of elements. So, does anybody have an example of a molecule off the top of their head? Anyone on Zoom who has an example of a molecule?
CO2,
CO2, what is that?
carbon dioxide.
Yeah. So we give off carbon dioxide when we breathe out, so maybe we all need to take a deep breath. I just exhaled a whole bunch of carbon dioxide. What type of organism really needs carbon dioxide?
Plants.
Plants, yeah. Let's see, is that, oh. Who keeps answering that for me? Good job, I want to give you a good job. Oh, hi, me. Okay, good. So that's an example. That's carbon dioxide. I'll do one more example of a molecule. H2O. Nice. And that is? It's water, yeah. All right, so H2O is water. These two examples were ones made up of different elements, but it can be made of the same atom or different atoms. So O2 is made up of just oxygen, and this is just oxygen. Right, that's what I just took a deep breath of. All right, so that's an example of how it can be made up of the same element. Okay, so we have different types of bonding. So now we're going to look at this like within the molecules or within multiple molecules, how do they bond together? So when you take a drink of your water, you're not having these, you don't feel these individual elements. You don't feel hydrogen and oxygen in your water. Like that would be really weird. So they have all these certain types of bonding that allow you to understand how things are put together, but on a bigger scale, you can't really see it. Okay, so ionic bonds, these are atoms in the same type of bond that donate or take electrons. Okay, so the way I remember this one, and this is super important, I remember this by saying, let's see here, if it lets me write. I want like a red pen. I remember this by saying, I donate. I'm going to write that on the board for the people in person. There we go. Look at my computer handwriting out there. So the way to remember an ionic bond is by saying, I donate. Okay, this is a little thing that I remember because I can donate. I can donate blood. I can donate my clothes to Goodwill, things like that. Because we're going to go through some different types of bonding, and we have to understand what do they do with their electrons when they bond together. Remember, those electrons are the ones on the outside. So if we have two molecules or multiple molecules coming together, maybe two different elements, how are they bonding with those electrons on the outside? The result in an ionic bond is a stable outer shell, and it occurs between particles that are charged. Okay, so if you think about it, laws of attraction, right? Opposites attract, positive and negative attract. So it says this occurs between charged particles or ions. And we'll talk about that in just a second. There's a good picture on the next slide that I'd like to show everyone. So here we have the ionic bonds. All right, we have a couple of examples, or it's the same example of ionic bonds. So we have a sodium ion, which is Na, and a chlorine item, which is Cl. You guys have maybe heard of sodium chloride, but what does that actually mean? So because of the amount of electrons that are on the outer shell of sodium and chlorine, sodium has a positive +1 charge, and chloride has a -1 charge. So when we put +1 with -1, it neutralizes and it makes it a stable ionic bond into what we know as salt, right? Salty. All right, so these are salt crystals. They're made up of sodium and chlorine, that makes sodium chloride. So we have the orange as the sodium, positive charge so if you ever see just a plus, it means it's +1. You don't have to put +1. If you see +2, then it means has a charge of +2. But with just the plus, it just means normal positive, okay? So it makes these crystals, salt crystals that we know and put on our French fries and our vegetables, right? Because we're all responsible and eat vegetables all the time. So that's an ionic bond. So we also have another type of bond. It is called covalent bond. And these are atoms in the type of bond where they share electrons. They result in a stable outer shell. So the way I remember this one I think I say co-share. So like you could share with your coworker or your things like that. That's kind of how I remember that. Co meaning share. Like a coworker means like you kind of share the same responsibilities at work or you work at the same place. So covalent bonds, they're going to share those electrons. It results in that stable outer shell. Both ionic and covalent bonds are the ones that result in that stable outer shell. Okay, we want them to be a stable outer shell because we want our salt to be something that we can eat, right? We want water to be something that we can drink. If it wasn't stable, there could be some difficulties with that. There's some different acids and and bases that end up not being stable, and it affects their acidity or their basic properties. And there's just some things you just don't eat or drink. You can drink milk, but you're not going to drink like sodium. Sodium bicarbonate is in Tums. I'm trying to think of another example. Like hydrogen peroxide, probably shouldn't drink that. Wouldn't recommend it. There's just some things that don't have stable outer shells. Okay, so here's a little bit more descriptive of that covalent bond. So we have hydrogen dioxide, which was a great example on the board. This is water. Hopefully you guys are drinking enough H2O, especially on these warm days. Today's pretty nice though. It's like 75 and sunny. Beautiful. Hopefully you guys can get outside. So here we have oxygen. Oxygen has 8 protons, 8 neutrons, and it has 1, 2, 3, 4, 5, 6 on its outer shell of electrons. We want it to have 8. Hydrogen has 1, so we add 2, and we make hydrogen dioxide. These electrons are just shared. They aren't necessarily given to one another. They're just equally shared in the middle to make a covalent bond. So when 2 oxygen atoms covalently bond, oxygen gas results. Oh, excuse me. Sorry, this was hydrogen dioxide. This is oxygen gas. That O2, that example I gave right here at the bottom, that is oxygen gas. So that's what you're breathing in and out. It's usually like if you've ever seen somebody who's on oxygen who has the nose tube, what's in that tank is O2. So here at the top, like I said, I'm sorry, I got my figures mixed up. Water is H2O. You've got 2 hydrogen ions and an oxygen ion, and they come together, they share their electrons to have a stable outer shell. All right, we're moving through here. So what are the properties of water? Okay, so we all know and have learned to love water, I'm sure. But we know that water is liquid at room temperature. You guys have known this since you were in elementary school. If you put water in the freezer, it's less dense than liquid water and it becomes a solid, right? So if you make ice cubes with water, the liquid water does not change temperature quickly. I think about it every time I'm trying to make some Kraft mac and cheese. Like you're like, please can this water boil a little bit faster? I'm getting hungry. Water has a high heat of evaporation. So if you ever just leave a pot of boiling water on the stove and it has a nice rolling boil, it's not going to boil over, and you just leave it there for a while, eventually all the water will evaporate. Molecules of water cling together. Like I said, this is really important because you don't just feel a whole bunch of molecules. It doesn't feel like marbles in your mouth when you're drinking water. The molecules of water itself cling together. And we'll talk about what kind of bond that is. And then water is a solvent for polar molecules. Those are a couple of terms that we don't necessarily need to know in this class, but just knowing those properties of water are super important. So molecules of water cling together, and that's through hydrogen bonds. I believe we talk about that next. So let me get that up on the screen here. So hydrogen bonds, every time I see the word hydrogen bond, I think it's weak. It's a pretty weak bond. Hydrogen bonds occur between a hydrogen in a covalent bond and a negatively charged atom. So we have a whole bunch of water molecules So this is how you show water, and this right here, that's a hydrogen bond. And that's how those bond together. Remember, we just talked about this one right here. This one was covalent. Okay, so let me draw that a little bit here. So we talked about this bond already right here. This is a covalent bond, people on the Zoom. And then right here, we've got this one is a hydrogen bond. Like I said, hydrogen bonds are relatively weak bonds. When you think of a covalent bond or an ionic bond, those are stronger than a hydrogen bond. All right, does anybody have any questions so far? Okay, hopefully this is like a little bit of a review of high school chemistry. We're just kind of briefly touching the surface, some basis to know some other things that we'll talk about later in this class. So acids and bases, that's a good little segue there. Acids are substances that dissociate and release hydrogen ions, and bases are substances that take up hydrogen ions or release hydroxide ions, OH negative. Okay, so that's like a lot of terms, but what does that mean? So maybe you've seen like a level of pH, how acidic or basic something is, and that is a scale of 0 to 14. All right, and we'll talk about that in just a second. 7 being neutral. Okay, this is a pH scale. And if you were to look up a picture of a pH scale, there's some pretty graphics and things like that of what a pH scale looks like, and we will show that in 2 slides from now. So what is that pH scale? So this is the measure of how acid or basic something is. It's the measure of the hydrogen ion concentration. So we just said if something, it has a lot of H pluses, it's going to be pretty acidic. Working scale between 0 and 14 with 7 being neutral. A pH below 7, so here is acidic, and above 7 is basic. So this is acidic all through here. Anybody know where water would fall on this scale? Has anybody ever heard of like pH water? When people get the pH of their water adjusted, it's very small, it's by a decimal point usually, but how about on Zoom? Does anybody know where water would fall on this scale?
Wouldn't it just be 7?
Yeah, Macy, that's right. It would be 7. Perfect. Yeah, so water would be right here in 7. Sometimes I've heard of people getting their water pH adjusted. I think they put it to a 7.2, but don't quote me. I'm going to have to look that up after class. So some people go and get like a certain jug of water, a certain like bottle of water and things like that, have the pH adjusted. It's supposed to have benefits to your body. I don't know much about that, but 7 neutral, that's where water falls. Would anybody know where like orange juice would fall on this scale? I guess the answer would be either be acidic or basic. Anybody on Zoom, where does orange juice fall on this scale?
Acidic.
Yeah, acidic. I'll just put some OJ. How about milk? Where does milk fall? Or like cow's milk? Yeah, Sam. Basic. Yep. Milk. Good. I give the example of, has anybody ever like heard of or done the challenge at Buffalo Wild Wings when it's like the hottest sauce of wings? I would never do that personally, but when you eat something really spicy, or maybe you're eating a jalapeno popper or you're things like that, or a really hot buffalo sauce, they tell you not to drink water to cool your mouth off. Has anybody ever heard of that before? They tell you to drink what?
Milk.
Milk. Yeah, they tell you to drink milk. It helps to neutralize that really acidic, hot, spicy, zesty taste. If you put water there, water's neutral. It's not going to neutralize anything if it's already kind of neutral. That milk, that basic, wherever that ends up being, it is going to help neutralize, not have that burning sensation in your mouth. Okay? On a scale of 0 to basic, I'm fairly basic. I like Starbucks. I walk my dog and drink my Starbucks, my AirPods in. I feel pretty cool. I'm pretty basic. Okay. All right, so here's a little bit better picture than the one I drew on the board. Okay, you can see that where things fall on this scale. Like I said, we don't really want to drink household bleach, which I think is what's in here. It might be. I think this is bleach water. Don't drink the bleach water, right? We want to drink pure water. Has anybody ever gone to the ocean and you crash in the waves and you get that really bad taste in your mouth and your nose? You don't really drink seawater either. If you're stranded out on the boat, you know, you're out at sea, you're like, but I'm surrounded by water. I'll never die. Don't drink seawater. Super, super salty. So you've got things like tomatoes, black coffee, urine, vinegar, cola, your stomach acid. Your stomach is fairly acidic. When it's acidic like that, it helps break down food that you eat. We have lemon juice. Hydrochloric acid is like very acidic. Don't ever touch hydrochloric acid. Hydrochloric acid, I did that in organic chemistry. It burned my fingers. That wasn't that great. Household ammonia, household bleach, sodium hydroxide. So that's just a couple examples of those things. So here's that acidic part, here's that basic part, here's the neutral, and then there's that H+ ion concentration, okay? You don't need to know a ton about this, but understand that as the concentration of H+ ions increases, so this is a really small number, right? To the -7th. That's a pretty small decimal. You probably can't really think about that in your brain right now. It's a little bit, it's Monday still, right? But as that H+ ion concentration increases, it becomes more acidic. And as the OH- increases and the H+ decreases, you get more basic. So that's just a brief overview of that. I don't normally do any test questions about the ion concentration. Okay, making and breaking down organic molecules. Okay, so we have a dehydration reaction and we have a hydrolysis reaction. Okay, so these are some scientific terms that kind of tell you what they are in the name, which sometimes that's pretty helpful. So this dehydration reaction, has anybody ever been dehydrated before? Maybe like every day if you don't drink enough water, but maybe somebody has actually been like seriously dehydrated, like I was when I was a really young kid. I think I was like 4 years old and I had to go to the hospital because I was dehydrated. So they pump you full of fluids. They need you to get the removal of water happened, right? So a dehydration reaction, the name is telling me that there's a removal of water. This allows subunits to link together into larger molecules. So when we get rid of some water, molecules are able to come together and make some sort of bond that helps us make some sort of organic molecule. And I'll give an example of that in the next slide. A hydrolysis reaction, hydrolysis, hydro. Does anybody have a hydro flask? I have a hydro flask. What do you put in your hydro flask? Most likely water, right? We got a hydro flask gang right here, yeah. So hydrolysis reaction is the addition of water that breaks larger molecules into subunits. So when we add water to some molecules, we can make some larger molecules. So here's a couple examples of this. This would be kind of a pretty good test question, homework question, like draw a hydrolysis reaction. Show me that there's a removal of water, you know, or a hydrolysis would be adding water. Dehydration reaction is the removal of water. So here's an example of that. So we've got subunit. A subunit just means that this is some sort of molecule. It has something on the other side. But what we really want to just focus on here is that here's a water molecule. It has two hydrogens and an oxygen. And when we release that water, we bring these subunits together to make whatever we're making. Here we have a hydrolysis, remember hydroflask. You add that water in to that subunit and it makes the subunit that we need. Okay, so the breakdown and synthesis of macromolecules. Excuse me. Okay. So what organic molecules are found in living organisms? So people probably recognize some of these terms because they're in the foods that you eat every day, right? Anybody know what a carbohydrate is? What's an example of a carbohydrate food? What's something that has carbs in it?
Bread.
Okay.
Bread, there was one more. What else? I see Barbara's mouth moving and I really want to know what you're saying, but I just can't hear you. I'm so sorry. You can type it. I'll look for it on the chat. Yeah, so carbohydrates, potatoes. Yeah, potatoes have starch in them, which is a carbohydrate. I'm also thinking of like some sugar, right? Glucose, all right, is a carbohydrate. Let's move on to the next one. So now we have lipids. Does anybody know what a lipid is?
Mm-hmm.
Yeah, Sam? Fat, yeah. So lipid is a fat. I think of there's good lipids, bad lipids. There's lipids that help with different things. You have lipids in your cell membranes, unsaturated versus saturated fats. And then we have proteins. Does anybody have an example of a protein or a food that has protein in it?
Meat.
Yeah, a lot of meat has protein in it. Yes. Legumes, that's a good one. Thanks. That was a good one. What else has some protein in it?
Peanut butter.
Peanut butter, yeah. So all the nuts, almonds, peanuts, they have protein in them. And then we have nucleic acids. You're like, whoa, I don't eat nucleic acids. That's weird. What's on this list? Nucleic acids are what make up your DNA. So deoxyribose nucleic acid is what DNA stands for. And I'll say that one more time. Deoxyribose nucleic acid. Nucleic acid. So that's what DNA stands for. And then you have RNA, which also is a nucleic acid. So these are all what we call macromolecules that make up living organisms. So we need all of these things to survive. So let's start off with carbohydrates. They are made up of subunits called monosaccharides. When I see the word monosaccharide, I start to look at what the word is telling me. So the word mono, it just means like one or small subunit. So when you think of somebody that says, yeah, let's go fight in the yard, mano y mano, that just means like one-on-one, right? So monosaccharides, so that's a subunit of those carbohydrates. Carbohydrates are made up of carbon, hydrogen, oxygen, in which the hydrogen and the oxygen atoms are at a 2-to-1 ratio. So there's always 2 hydrogens to 1 oxygen in a carbohydrate. They function as short and long-term energy storage. So if any of you were an athlete or like to work out and are athletic, you'll know that sometimes your sports teams or your coaches will say, oh, you got to carb up before your long run tomorrow. Okay, yeah, there's some studies that show that carbohydrates are good for long and short-term storage, but then the debate is like how far away from your competition you should do that. But what it's saying is carbohydrates, that long-term energy storage and that short-term. So does anybody have an example of like what a short-term energy storage carbohydrate would be? Yeah, so Nick in the class said candy bar. So if you have a candy bar, like eat a Snickers, you're a diva when you're hungry. It's gonna help you for a little bit, right? You're not gonna be a diva for too long, but then a little bit after that, you're gonna probably have a sugar crash, right? So it's that short-term energy storage. It gave you some energy, gave you a little bit better attitude. Maybe you like chocolate. That would be a short-term energy. A long-term energy storage, anybody have an example of a long-term energy storage carbohydrate? How about from Zoom? Long-term energy storage carbohydrate. Example.
Pasta.
Pasta, yeah, especially if you get like a whole grain pasta, things like that, or maybe like a veggie pasta, things like that. those are going to have long-term energy storage. So you're not going to have like that sugar crash. You're going to have some like standing energy for a while. It doesn't mean that it's going to last forever, but it'll be a little bit more sustainable. And then they can be found as simple or complex forms. Thanks for your participation. So what are some simple carbohydrates? So we have a monosaccharide, which I talked about. Remember, mono, one-on-one. Monosaccharide, it's one carbon ring as like found in glucose. So a carbon ring is these hexagons that have carbons on them. Your body, the shape of them in your body is like this, but you don't just have green hexagons floating around in your body when you eat carbohydrates and things like that. This is just how, when you think of the itty bitty molecules, how the carbon and the oxygens and the hydrogens are formed. So when they say a carbon ring, it's just like a ring of carbons that happen to be in a hexagon form. And then a monosaccharide example would be glucose. That's, what does it say? C6H12O6. At one point I had to memorize all of those, but sometimes, you know, when you go to memorize for a test and then you have like all these C12H6, whatever, all of those memorized, it's gone over my head. But what you need to know is that monosaccharide is a single carbon ring. It's just one of those rings of carbons. And then you have a disaccharide. This is two carbon rings. It's found in maltose. Does anybody know where you can find maltose? This is a good. So it's not in a chocolate malt, but maltose can also be found in some malt beverages. So like some adult beverages that are considered malt beverages have maltose in them. So that's a fun fact. We will move forward to a polysaccharide. So these are some complex carbohydrates. So poly, when I see the word poly, I think of the word many. They both have a Y in them, if that's a way you remember it. But when I see polysaccharides, I just think a lot of those carbon rings. So these are carbon rings that are made up of many carbon rings. Glycogen is a storage form in animals. So glycogen would be a carbohydrate that would be found in a lot of meat proteins. Starch, like Barbara's example, which is in a potato, is another example of those polysaccharides. So if you had to take a guess out of this slide or the last slide, let's say for this slide, is this the long or the short-term energy storage? This slide right here. Choosing between long and short. How about Zoom? Is this a long or a short-term energy source, this slide that we have pulled up?
Long.
Yeah, Betsy, this is a long one. So like when you have a meal that's got like some, you know, you're feeling good, you're having some meat protein, and then maybe you're gonna have some roasted potatoes with it or some French fries or whatever. Those are gonna be a little bit longer energy source than in the last slide with glucose, which is just sugar, right? Maybe than having like a muffin, even though muffin can be pretty good meal, things like that. You can have it for breakfast sometimes. But you're going to find a lot better energy source, like your pastas, your potatoes, are going to be these complex carbohydrates that are going to be made up of polysaccharides, which are more than one of those carbon rings. If you think about it a little bit, if you've got a lot of those carbon rings, it's going to be a lot longer to metabolize that than it is a simple or two monosaccharide and a disaccharide, how that metabolizes. Good. We are trucking forward. What are lipids? So lipids do not dissolve in water. So when I think of a lipid, maybe you've seen a picture or a video or something of just what a piece of fat looks like, even from the human body. I think of just a piece of fat, just throw it in a bucket of water, what's going to happen? It doesn't dissolve in the water. Sodium chloride dissolves in water, right? Salt water, you can mix that together. But lipids are really important for a lot of different reasons, one of them being energy. There's some differences between good and bad lipids. They're found in the cell membrane, and this is super important. and we'll talk about it a little bit more in detail, probably Friday, cell membranes, but they can be found as fats and oils, phospholipids, and steroids. Okay, so fats and oils, I think of your vegetable oil, olive oil, avocado oil. These are all lipids. If you dump some avocado oil in your boiling water before you make some pasta, I don't know, some people put oil in their water before they boil it, you'll see that there's little oil droplets. They don't mix together. But then you add some salt into your boiling water before you make your pasta, and the salt dissolves to an extent, right? If I had this much water and I put a pound of salt, obviously to an extent it doesn't dissolve every single time, but when there's an equal or more greater water volume than there is salt concentration, it does dissolve. So fats and oils don't dissolve in water. Phospholipids are what's found in that cell membrane. Steroids, we'll talk about that here in a second, and how they're important as well. Maybe you've heard of some steroids probably in the news, what athletes use, or maybe you use it for, I've heard of it working on some rashes and things like that. So fats are usually of animal origin. Okay, so your butters, your whole milk will have some fat in it, because the percentage of the milk is just how much percent of fat they leave, right? So you have skim milk, which is not going to have, or fat-free milk is not going to have any fat on it. 1%, 2% is the percentage of fat. Whole milk, they keep all of the fat on there. It's solid at room temperature, right? Don't you kind of wish fat was liquid at room temperature? Am I right? The function for long-term energy source, the isolation from heat loss, and cushion for your organs. Okay, so there's usually a negative connotation with fat, but also you have to think of if you were to, God forbid or something, trip and fall on the sidewalk, right? You're a little thankful that you've got some cushion around your internal organs. Like you don't want your heart just kind of like sitting here next to the skin and then you trip and fall. Like it'd be kind of dangerous, right? So I know that's like that's a very morbid example. I could have thought of other things, but just essentially that fats are good for cushion for those internal organs. It's also good for insulation from heat loss, right? You go outside in the middle of December in Iowa and it's 30 degrees, or maybe it's negative 30 degrees, who knows, maybe it's January. You're so glad that you're covering up some of those internal organs with some fat, or else your body would kind of, start to shut down because you're not protecting those internal organs. Think back to, what's your main goal for your body? Your body wants to pump blood and breathe and live, right? That's not your consciousness, but your actual body. So by having some form of fat over your body, it does protect from heat loss and the cushion for those internal organs. Then you have oils, usually from the plant origin, which is the example of like your avocado oil, your vegetable oil, your olive oil. Did I already say that? Yeah, all of those different oils, you may use them for cooking. They are liquid at room temperature, right? When you go to grab the jar of olive oil before you, I don't know, what do you guys do with olive oil? Roast it for potatoes or dunk it in some bread or things like that. The oils are liquid at room temperature. So when I think of the two different things, I think of an example of fat, like if you're going to have a piece of steak, unless you're vegetarian or vegan, I apologize for this example. But you have the fat sometimes on like a piece of steak. That's solid at room temperature. Then you have oils that may be like I said, you use for cooking or things like that. Okay, what is the structure of fats and oils? So it's a glycerol molecule and 3 fatty acid tails. So the way I like to look at it is it kind of looks like, another example would be kind of like this with 3 fatty acid tails. Okay, I'll draw that here on your guys' slide so you can see. I drew something like this that has 3 fatty acid tails. Okay, so you have that glycerol molecule and it is right here. Remember, glycerol is a type of what? Does anybody know what glycerol is a type of? It's a carbohydrate, right? Because we have carbons here. We have a 2-to-1 of your hydrogen to your carbons. Then we got 3 fatty acid tails. Okay, so this is what that structure of fats and oils looks like when you have a dehydration reaction. So here's some H2Os right here. And for you guys on Zoom, here's the H2Os right here that I'm looking at. When we go to a dehydration reaction, remember that we are removing the H2O. Sorry, that was a really bad 2. There we go. We remove those, and then we have a fat molecule. If you want to break these molecules down to a glycerol and a fatty acid, you add water. So that hydrolysis reaction, we want to add H2O into that. There we go. Good. Okay, so understanding fats when reading a nutrition label. So we will have an assignment that'll be outside of class. It'll be, I think it's due next week. But again, I'll check that date. I should check the dates before I tell you these things. But where you'll have to either get a food label, maybe from your dorm room, or find one online, a picture of a food label, and kind of interpret it and read through a few different things to understand it. So when it says that the recommendation for total amount of fat for a 2,000 calorie diet is 65 grams. So that means that on the average person, regardless of age or gender or things like that, weight, average recommendation of calorie intake is 2,000 calories. Now, there's so many other things that go into determining that. It does not mean that for every individual you should have 2,000 calories. Some athletes, I mean, I think of the example of like Michael Phelps. Does anybody remember how many calories Michael Phelps eats in a day when he's competing?
10,000?
10,000. I'm like, man, that's a lot of Olive Garden breadsticks, but I'm willing to take the challenge. So there's obviously different activity levels, different life goals, things like that. So it doesn't mean that I'm sitting here saying eat 2,000 calories a day, but on average, that means that you should have about 65 grams of fat. Okay, and that doesn't just mean like, yes, more potato chips. Oh, I love potato chips. There's good and bad parts of fat, right? So be sure to know how many servings there are when you're reading a nutrition label. And then the percentage DV of 5% or less is low and 20% or more is high. So the daily value is what DV stands for there. If you have 5% or less of that, you would consider to have low fat intake, and 20% or more of that is high. So 5% of 65 and 20% of 65. So you try to stay away from trans fats. Is it okay to have trans fats once in a while? Yeah, absolutely. Please eat your potato chips. But it doesn't mean that you should have a bunch of trans fats. They're harder for our bodies to break down. And it says, would you eat the food on the following nutrition label? Why or why not? So the next slide, there's going to be a picture of a nutrition label, and we're going to talk about whether or not we should eat this. Okay. So give it a moment. Check what we have here. We're checking understanding. Okay, so it says to start here. Perfect. We want to know how many servings per container. So the serving size is 1 cup and there's 2 servings per container. So hopefully you guys know that if you wanted the total calories, you take this times the number of servings. So it would be about 500 calories if you ate the whole thing, the whole container. The number of calories from fat is 110. All right. So you're supposed to limit these nutrients. Okay, those are the total fat, the trans fat, the cholesterol, and the sodium. Some diets actually require a lot of sodium. I can think of some individuals in my personal life who are supposed to have an increased amount of sodium, depending on what their body needs. But these are the ones that tend to give you heart troubles, weight issues or things like that, but they're good in moderation, right, as is anything. So this one says it has 12 grams of total fat, and it's 18% of your daily value. So it's 18% of those 65 grams we talked about. Then you've got, of that fat, 1.5 grams is trans fat. It has 30 milligrams of cholesterol and 470 milligrams of sodium. And And then the bottom part is just saying you should get enough of these nutrients, right? You should look for things that are nutrient dense. And those are your vitamin A, your vitamin C, your calcium, your iron, which we talked about. Fiber is pretty good. Protein, but notice how it didn't really put a bracket on sugar. That's, where do you guys think sugar would land on this? Probably a little bit towards the top is what I would say with limiting, but there's different types of sugar, right? There's the sugar that you get in a Snickers bar, and there's a sugar that you get when you're eating a banana or some orange slices, right? So there's different types of sugar. So the question was, should we eat this food? Well, everything is good in moderation, but what I would suggest is that this is pretty high in fat and sodium and cholesterol. As you can see, there's less dietary fiber, there's no vitamin A, barely any vitamin C, but it has 18% of your fat, and it's about, 20% of your daily sodium value, but, it's good and bad. And my class last term said, well, what is it? And I'm like, actually, I don't know. I have no idea what this nutrition label is for, because it maybe would determine whether or not we should eat it, but I don't really know what this belongs to. So we're going to move on. Okay, so what is the structure of a phospholipid? This is a great test question because this involves two types of things. It involves two chapters, right? We're going to talk a little bit about cell structure and function in the next lecture, chapter 3. And this is showing the type of structure that makes up that phospholipid. It has a polar head and non-polar tails. So just to give you a little brief moment of reference point, like where are we at? So right now we're going to be looking at an animal cell. And the reason I know this is an animal cell is because plant cells have rigid cell walls and animal cells do not. So this is an animal cell. It has a cell membrane. This is the inside of the cell right here, and this is the outside of the cell right here. Okay? The cell membrane is made up of a phospholipid structure. It has a semi-permeable membrane, which means it allows some things to go in and some things to not go in. Okay? And we'll talk about what is allowed to go in and out of that cell membrane. And it's because of those polar heads and those non-polar tails. So polar and non-polar, remember we talked about water being a solvent for polar molecules? So water can come in and out of your cell membranes, so that's pretty good, right? We need some water to do that. Alcohol can go in and out of your cell membranes. That's why there's a physical effect when you drink alcohol. It also says one fatty acid is replaced by a polar phosphate group. Phospholipids are the primary components of cellular membranes, so that's what I talked about. I also think of your semi-permeable membrane like the bouncer at a club, like somebody's sitting there like saying, yeah, you can go in, you can't go in. It just depends on whether or not it's solvent, it's polar or non-polar. These are all the things that will help determine whether or not things come in and out of our cell membranes. Now, there are some things you probably don't want to come in and out of your cell membrane, and we'll talk about that as well. The next slide says, what is a steroid? You've probably heard of steroids before. You probably I think of like these bodybuilders way back in the day, or maybe even nowadays, and there's more strict rules about this of helping you build muscle and get large, like steroids. But steroids aren't just the getting large steroid injections that you think of. Steroids can also be something topical that you use on like a poison ivy rash or things like that. The structure of a steroid is 4 fused carbon rings. So there's 4 carbon rings in that picture on the right hand side on the top. That's a picture of a steroid. Other examples of steroids are cholesterol and sex hormones. So testosterone, you've maybe heard of that, and estrogen, those are different sex hormones, and those are considered steroids. They are considered types of lipids. So that would be a couple examples of those. Moving on, I'm trying to push through so we can get done here today. We've probably got about 9 more slides, including this one. So sticking with me. I know the Zoom people, you guys maybe had Zoom all day today or a little bit last week, so hoping to keep your attention. If you need to stand up at any time, that's up to you, because we're recording this anyways. So what are proteins? They're made up of subunits called amino acids. And subunits is a word that we keep using in the last couple of things. Subunits are essentially like the Legos, the building blocks, are what going to make up these units. So the subunits of carbohydrates are monosaccharides. When you have more than one monosaccharide, you have two. That's a disaccharide for carbon dioxide. Carbohydrates, excuse me. The proteins are the subunits, the building blocks, the Legos are amino acids. You've maybe heard of amino acids before. And we'll maybe, on the next slide, we'll show a couple examples of amino acids. When you think of protein, like we talked about, we think about like meat protein, we think of legumes, we think of beans, we think of nuts that have protein in them. Eggs are one of those examples of a complete protein. They're going to have all, I think, 16 types of amino acids in them. Probably somebody's going to quote me and say that I'm wrong on that number, but eggs are a really good source of protein because of how many of those simple amino acids that are in there. Proteins are important for diverse functions in the body, including hormones, enzymes, antibodies, and transport. Some pretty important things. So hormones affect more than just estrogen and testosterone. Hormones can be epinephrine or no epinephrine. They can be serotonin. Does anybody know any other examples of hormones? I kind of gave the most. Melatonin, yeah, Barbara, that's correct. So maybe you take, or some people may take a melatonin before going to sleep. Is anybody, that would help you kind of drift off into a sleep a little bit better. Melatonin doesn't make you sleep. Melatonin helps relax. It helps relax muscles. So that's a great example. Good job. So enzymes are a little trickier. So enzymes are going to help break down things, and we're going to talk about what types of, there's different types of enzymes. Antibodies, that's kind of a trigger word right now. If you have the coronavirus antibody, maybe you've heard of that word, that matters these days. Antibodies are what help fight off foreign objects in your body and infections and things like that. And then transport, so there are cellular processes within your cell and within going from cell to cell that use protein carriers to do those different processes. So as you can see, proteins have a lot of different uses. You've probably heard of, like, if you are trying to like repair muscle, like after a workout, they say, go have a chocolate milk, go have a, some grilled chicken, or go have some black or pinto beans or whatever with something with protein in it. Why? Okay, so after a workout or after doing something like that, along those lines, maybe you went for a run or maybe you were lifting, you're essentially doing micro tears in your muscles. And what we want to do is then repair those muscles to be bigger and better after each workout, right? And then maybe sometimes you feel a little sore. But if you add some protein into your diet, your body is using that protein for the transport of different molecules to help mend those muscles. So that's why we need protein in our diet for that. They can denature and undergo a change in shape that causes loss of function. Okay, the best example I can give of this is an egg, right? When you add heat to an egg, it goes from liquid to solid, right? If I want to make some scrambled eggs, I'm going to crack the egg and I'm going to put it in a bowl with a little bit of milk and some salt, whisk it up. Maybe some people don't use milk. That's cool too. My boyfriend doesn't use milk in his scrambled eggs. It drives me nuts. But you whisk it up, you throw it in the, a frying pan or things like that on the stove, a skillet, whatever have you. And when you add heat to that egg, it goes from a liquid to a solid and something that we can eat. I'm not going to eat a raw egg. Some people can. That's totally fine for you. But that denature, it just means that it undergoes that change in shape and a loss of function by the addition of heat. So this next slide is showing some examples of amino acids and what they look like. You've maybe heard of some of these, you maybe haven't. I won't have you memorize these. I would never say list 10 amino acids because that's organic chemistry. That's cell structure and function. We're not cell structure and function. We're drugs and personal health, right? But we need to know kind of the idea of what amino acids look like, maybe a couple of words that you could recognize, and maybe understand what they are in context of other things. So valine, glutamic acid, lysine, these are all examples of amino acids. They have the option to be ionized, which means that they'd have some sort of charge. They could be polar or non-polar. And so these are just a couple of structures of those amino acids and what they look like. That is figure 221. Okay, so this is also another great test question. What are the 4 levels of protein organization? I've had students answer the question of draw a picture of this. And the next slide will be drawing a picture or showing a picture of what protein organization looks like, okay? Following along with me, I'm going to give like a metaphorical example, but hopefully it'll help. I don't have a loose leaf piece of paper right now. Here, we will use this napkin as an example, okay? So protein levels of organization, we talked about that the subunits, the building blocks for proteins are amino acids. So when we have the linear order of amino acids, we have that primary structure of proteins. Okay, so this just means that we have one little thread of like essentially little Legos of amino acids. I think of it as like a fiber that makes up this napkin or a piece of paper. Oh, somebody drew on it. Cool. So there's little individual fibers that make up paper, right? You guys know that. There's like little itty bitty strings looking things. That would be a primary organizational level of protein. Then we have the secondary, which is that localized folding into the pleated sheets and helices. So now we've got a whole bunch of those fibers, right? A whole bunch of those fibers, and it makes a piece of paper or a napkin, right? So this piece of paper is secondary structure. The fibers within this napkin or the piece of paper is that primary structure. So we're kind of organizing and going step by step. Tertiary is that 3D shape of the entire protein in space. Okay, so now we've got this right here. This is tertiary structure. Okay, this is that crumpled up piece of paper. 3D now, it's not flat. Right, flat would be as if it's on a piece of paper and put a whole bunch of fibers. Now we're 3D. We are tertiary structure. Primary, secondary, tertiary. Essentially means 1, 2, 3. And then we have quaternary structure, which would be more than one of these together. Okay, so here's that kind of example. Hopefully nobody actually uses these napkins. I probably should like throw them away. But all proteins have primary and secondary and tertiary structure, but only a few have that quaternary structure. So no matter what, most proteins are organized like this. But it's a little bit more rare to have more than one protein that's together, like this, to have that quaternary structure, that combination of more than one polypeptide. And the slide on the next picture will give you a better understanding of that. So what do the levels of organization for proteins look like? So we've got that first one. It looks like beads on a string, kind of that top one. So that's just like a string with some Legos on it or some beads. Those beads are the amino acids. And that's that primary structure. You can see there's the amino acids. The amino acids are held together by peptide bonds. And like I said, it's that sequence of amino acids. Then you've got your secondary structure, which is broken down into helices and pleated sheets. Okay, so a pleated sheet is kind of just thinking like if you were to fold your bed sheets, that's how I kind of think about it. A helice or a helix is like a spiral staircase. And that secondary structure is essentially a bunch of those beaded strings folded or curled up on each other. Okay, then we have and it kind of makes it look like a ribbon, essentially. So now we have that tertiary structure. So it's all the beaded string, pleated sheets, and helices. It's kind of hard to say. It's a tongue twister. It makes that tertiary structure. And then you have, lastly, the quaternary structure, which are the different colors. So there's more than one of the tertiary structure. And so that's just another example. The way I would word this on a test or a quiz would be, what are the 4 levels of protein organization, and draw an example. So you would have to essentially kind of draw this slide in your own way for your own understanding and be able to say, like, this is primary, this is secondary, this is tertiary, this is quaternary structure. Okay, getting towards the end. Here we go. What are nucleic acids? They are made of nucleotide subunits. So another great question for a test if you're looking at like the macromolecules is to name each of the subunits, right? We talked about carbohydrates, proteins, lipids, and now nucleic acids, and they're all made up of some sort of subunit. They're all made up of some sort of building block or Lego that'll help kind of make them into the structure that they are. And hopefully you're understanding a little bit that the organization, all the way from these elements and molecules, is we're building and building and building in order to understand the bigger structures that they are. Nucleic acids function in the cell to make proteins. DNA and RNA, their job is to make proteins, and those proteins are encoded from the DNA's code, right? And then those proteins are coded from DNA. They are in charge of different functions. Okay, so, and there's a lot of them. In graduate school, they told me exactly how many proteins we have, and I think it was like 20 or 30,000 specific proteins, and we only know like what 1,000 of them do. Those numbers may be off a little bit, but we don't know. We know that these proteins exist, but there's so many that we just don't even know the function of. And our DNA encodes for those proteins. And that's why I said earlier it includes RNA and DNA, ribose nucleic acid and deoxyribose nucleic acid. That's what DNA and RNA stand for. So we need DNA and RNA. You guys know that DNA is what makes you unique. It makes you unique from your siblings and your parents and your cousins and your grandparents, and things like that. So what are the 3 parts of a nucleotide? Okay, a nucleotide is the building block of those nucleic acids. The 3 parts are the phosphate, the sugar, and the nitrogen-containing base. This would also be a great little test question where you could draw your little picture and tell me about what, those subunits and things like that. A phosphate is an element. It's there indicated in yellow, and then you have the sugar. We talked about sugar. Sugars are carbohydrates, right? So we've got that carbon ring. It's green. I think that the PowerPoint does a good job of showing the colors, and they're very consistent with that. And then you have your nitrogen-containing base, and that's here. Those are the parts of the nucleotide. Nucleotides make up nucleic acids. So what are 5 bases found in nucleotides? Maybe in high school science, you were given a string of letters, and then they made you do the pair of letters underneath that, I'll show you, help make up the DNA rungs of the ladder. Okay, pause there. It's okay if you don't know what I'm talking about. There are 5 bases found in nucleotides. There's A, G, and C, T, and U. Okay, adenine, guanine are double-ringed purines, and cysteine and thymine and uracil are single-ringed pyrimidines. And these are in DNA, A and T pair together, and C and G pair together. And the way that I remember this is A and T are both upright letters, and G and C kind of both have like a curved part to them. So that's how I remember that A's and T's pair together and G's and C's pair together. So let's talk a little bit more about this. So you probably know that DNA is a double helix. It's a spiral staircase, and it's twisted up on itself. The spiral staircase is super tight and spiral to protect it. You don't want your DNA to be damaged by any means, because it could code for different things. But it's a kind of like a spiral staircase, but the steps of the staircase are these bases. So you have an A and a T, a C and a G, a T and an A, a G and a C. And they pair up together in the middle, and those are considered nucleotides. And at a certain area in your DNA, having a mix of these letters in a different pattern is what codes to make us unique. So maybe instead of this being a T, it was a G, and just that one change right there could affect the type of hair you have, whether it's curly or straight or coarse or thick, or maybe the color of your hair or the color of your eyes or things like that. So the coding of these nucleotides is what really affects the uniqueness of the DNA. It also is fairly important that we have a phosphate backbone. So this part right here, the ribbon-like structure, is made up of this sugar phosphate backbone. I think of it as like the railing of the spiral staircase. Like if I was walking down, I'd be hanging onto that sugar phosphate backbone. It's protecting me. It's helping me walk down those stairs, things like that. Because I can be clumsy sometimes. All right, so here's a summary of DNA and RNA and the structural differences. Great question, right? Like draw a Venn diagram of what is the difference between DNA and RNA. They probably have some similarities, right? And they have some differences. So the first difference is the type of sugar that they have. So sugar in RNA is a ribose, and DNA has the sugar deoxyribose. And it's just in the title of their name, right? Deoxyribose nucleic acid, ribose nucleic acid. That's a difference between them. You've got bases A, T, C, and G in DNA, but in RNA, you don't have the letter T, you have the letter U. So that's a difference there. DNA is double-stranded, while RNA is single-stranded. And RNA helps create those the coding for proteins, while DNA codes for RNA. All right. Here is just a summary, if I can pull it up on here. A summary of those macromolecules. This picture is in your book, I believe. And if not, you guys have access to this book picture, and you'll have access to this PowerPoint on my Wartburg. Okay, so there's 4 macromolecules: carbohydrates, lipids, and proteins, and nucleic acids. These are the macromolecules that make up the organic molecules that make up living organisms. In the second column, you have examples. The third column is monomers, essentially those subunits. What are those building blocks? What are they made up of? And lastly, the function. Okay, we talked a little bit about all those functions. We talked about carbohydrates, having that short-term versus that long-term energy source. It also, we've now found out that it's also good for structural things for those molecules. So that sugar phosphate backbone, sugar, right, in that DNA, the sugar phosphate backbone is that railing. It helps the structure of the DNA. You've got lipids, those are those long-term energy storage. They're also in the cell membrane. You have proteins, we know that they're good for support, metabolic reasons, transport, regulation, motion. And then we have nucleic acids. They're the storage of genetic material. It's what makes you unique, is your DNA. And that breaks down to nucleic acids and bases, those nucleotides. All right, what questions do people have about that? Is there anything? Zoom, how did we do? The audio was okay? You could hear me talk? That's so great. Honestly, we had more problems in person than we did online, right, with the PowerPoint, so that's pretty good. So like I said, Chapter 1 assignment is due tonight at 11:55 p.m. If you don't know where that is, if you can't find it, email me ahead of time. Okay, Chapter 2 assignment will be due on Friday, same time, same place. And then I believe, let me pull this up quick. Syllabus. So we have food label homework. I'm going to hand that out or email it on the next class. I'll email it when we're done here in class. It'll be due September 7th, which is Monday. I'll email it out. I'll give you guys an outline, a summary of everything I just talked about. September 7th, that'll be a food label homework. It's kind of like a worksheet more so. And it'll have a couple of questions on there that'll say like, draw this structure. If you don't have access to a printer and you can't print that off, just find a picture of it on the internet and show me that you understand it, maybe with some text underneath it if you can't print it off and draw it. And then the other part of it is that you find a food label and you interpret some of the things on that food label. It has you do 3 different food labels. And what we'll do is if you are in person and you feel comfortable turning that in, you can just staple those food labels to the outside, whether that be like a box of mac and cheese or a granola bar or whatever. Or if you don't have access to that and you'd like to turn it in online, you can find pictures of food labels when you Google them online. You could say food label for veggie straws and it'll pop up, and you can just take that Google image and put it on your assignment. Okay, and I'll email that out as well. Any other questions in person, people? My friends in person, yeah.
I couldn't find the email about the lab. Is it just like we go first and then like the first half of the last names go first?
Yes.
It's the same thing. Okay.
So the question was, he couldn't find where you figure out whether you're first or second for lab. So you guys are pretty much group A because you're the first half of the alphabet. So you guys will be here at 1:15. You'll be done at 2. 45 minutes in lab. Second half of the alphabet will be required to do that quiz, and then they will be here at 2:10, and they will go till 2:55. In between that, I'll just wipe down everything, restart, clean it up, and things like that. If you are one of my people who can't be in person at all, I will just log on to Zoom as normal, just like this. You can listen to me and watch as people do the lab. You'll have the lab instructions in front of you, and you're still required to do the quiz. You just read through the instructions, you follow along on Zoom for one of the sections, not both. I would recommend going to your regularly scheduled. So if you were scheduled to be first in person, log on to Zoom for that part, for the first 45 minutes. Watch us do the activity, follow along in your instructions, and then your quiz is also due at 11:55 on the same day. All right, anything else? Okay, well, in-person people, you guys are free to go. Email me if you have questions. Zoom people, if you have any questions, you can stay after. Other than that, it was nice to see you guys. And hopefully this went okay. It went all right. I'm trying to think. Let's see here, I'll stop.